Virologist Eugene M. Johnson framed the threat of this pathogen with a specific comparison: the destruction that HIV takes 10 years to inflict on a human body, Ebola accomplishes in just 10 days. The pathogen belongs to the Filoviridae family. This specific viral classification covers two main species capable of causing severe hemorrhagic fever in humans: Marburg, which carries a 25 to 30% mortality rate, and Ebola. It operates entirely apart from the biological mechanics of common human illnesses. It has no relation to influenza, mumps, rabies, or the varicella virus. It is an ancient organism tied to a specific environmental reservoir, deep within the rainforests of the African continent. Epidemiological tracking points to specific isolated environments, such as the Kidam Caves, where the virus circulates quietly among local wildlife. The virus achieves its lethal efficiency through a hyper-efficient physical shape and a direct mechanical attack on human structural integrity, despite its minimal genetic code. A single Ebola virion measures roughly 80 nanometers across and extends between 800 to 1,000 nanometers in length. This micrograph shows the virus's distinctive structure. Instead of a typical sphere, it forms long, branched filaments, frequently twisting into U-shapes or the figure 6 shape you see here. It is an enveloped virus. Its outer lipid layer contains 7 nanometer surface spikes that latch onto host cells. Beneath that envelope lies the machine's core, a tightly coiled helical nucleocapsid. Inside that protective helix is the payload, a single negative-sense RNA strand. The entire genome is remarkably compact. It is only 19 kilobases long and contains the instructions to synthesize just 7 known structural proteins. This minimalist blueprint contains the specific instructions required to outmaneuver the human immune system and trigger fatal systemic hemorrhage. This chart plots 5 Ebola subtypes. 4 share a common transmission vector, bodily fluids. Ivory Coast has 0 mortality, while Bundi-Bugoyo kills 25%. Sudan escalates to 60%. At the apex is Zaire, exceeding 90% mortality. Its lethal dose is just 500 virions. The 5th subtype, Ebola restin, is a biological outlier. In macaque monkeys, it is universally fatal, commanding a 100% mortality rate. In humans, restin causes 0 mortality. Yet it possesses a trait the other 4 lack. It is the only Ebola strain proven to be airborne. Minor genomic variations between these 5 strains dictate whether the pathogen acts as a harmless aerosol or a fatal fluid-borne disease. Once Zaire crosses into the bloodstream, it begins a targeted teardown of the body's internal architecture. The virus aggressively attacks and destroys human collagen tissue. It is highly selective in its destruction. While the virus erodes the softer structural tissues, it entirely spares the bones and tendons of the host. It also exhibits a specific biological tropism, showing a distinct preference for attacking the reproductive organs, heavily concentrating in the testes and ovaries. Collagen is the primary physical scaffolding of the human vascular system. As the virus deletes that scaffolding, the walls of the blood vessels lose their structural integrity. Ebola systematically dissolves the biological glue that holds the human circulatory system intact. The clinical presentation of this cellular collapse moves on a precise, unyielding timeline, though it begins quietly. For the first 6 days, the patient presents with standard, unremarkable symptoms, primarily a severe headache and a persistent high fever. Day 6 marks the inflection point. As internal collagen depletion reaches critical mass, the patient begins suffering from severe vomiting and bloody diarrhea. With the vascular system actively disintegrating, massive internal and external bleeding cascades throughout the body. Blood flow to critical tissues is choked off, triggering widespread necrosis of the internal organs and resulting in total multi-organ failure. The combination of systemic organ failure and uncontained hemorrhage leaves the patient in a state of profound physical devastation. Death occurs strictly between day 7 and day 15. That brutal 2-week timeline is simply the macroscopic symptom of the microscopic destruction of the body's connective tissue. Identifying the pathogen requires a precise chain of laboratory diagnostics, starting with standard microscopy and escalating quickly to RT-PCR testing. Advanced confirmations rely on assays like ELISA and radioimmunoassay. eventually moving to next-generation sequencing to definitively map the exact 19-kilobase genome. But high-tech diagnostics rarely match the reality on the ground during an outbreak. Historically, contaminated needles in under-equipped African hospital facilities were a primary driver of the virus's spread. Handling these 5 strains in a laboratory setting triggers the highest biosecurity mandates in the world, requiring maximum containment facility protocols. When an inoculation of just 500 microscopic particles guarantees complete systemic collapse in a matter of days, the only viable defense science can offer is a physical, impenetrable wall.